Okay, so good morning, good afternoon, everyone. Um, so in this meeting, so we have the privilege of having Mads Albertsen and Emil Sorensen to talk about or just Q&A between the two of us, between them and their experience and what we're trying to do. Just a quick background about accessible genomics, Mads and Emil. So what we have been trying to do is to try to bring genomic SARS-CoV-2 surveillance in areas where it's not yet present, particularly in the developing world. So what we've been doing in the past year was to partner with a lab in the developing world, particularly Philippine Genome Center in Mindanao, Philippines, to prototype. How do we actually do this? So we brought in a Minayan, we brought in the reagents, and also training for them. So last March was the first time that any organism was sequenced in the region of Mindanao, Philippines. And I believe this is also the first time that sequencing was done enabled by an international volunteer team. So our objective is to share this experience to other labs, so sort of create a playbook for them. And right now, we just actually today we just launched our crowdfunding campaign, and we're hoping to raise more money so that we could help other labs here in the area by also providing the training and the sequencer and the reagents. So when we saw that what you're doing, like producing 1,000 genomes a day, is like sort of mind blowing, and so we just love to hear about what you're doing in Denmark.
Sure. So a bit background of what we've been doing in Denmark. So back in March 2020, Denmark closed down, and at that point, I had a really nice group of scientists, around 10 people, including me, who were working a lot with nanopore sequencing, but on bacteria. And we thought, hey, can we use our expert knowledge in I would say, large-scale nanopore sequencing to help with SARS-CoV-2 surveillance in Denmark? And basically, within a few weeks, we got outreach to the hospitals and to the government lab who did it in Denmark. And I basically said, if you can ship me the samples, I will sequence them. It has to be said that in Denmark, we are extremely privileged. So I would say I have unlimited funding, basically. And I have one of the best teams in the world to do nanopore sequencing. So that meant we could get going very fast. On top of that, Denmark have an extremely well-functioning healthcare system. So every person is registered, everything is centralized, so we can actually get samples. I think that's once you solve the money problems, the biggest problems is always getting samples, as far as we can see. And in Denmark, there's no problem. So we probably have one of the largest test capacities for SARS-CoV-2 PCR tests in the world. We do 200,000 tests a day in Denmark, and we have a population of 5.6 million. So that's a lot. So in Denmark, we are very privileged to be able to set this up. And what we do right now is we sequence every positive sample in Denmark. It's actually sequenced with genomes. So around 90% to 95% of samples we get our hands on. And in the end, we have genomes around 70 to 75% of all positive samples in Denmark. It's set up really like a factory, I would say. So we have some really big PCR screening facilities in Denmark. Once a sample is tested positive for SARS-CoV-2, it gets picked out by robots and actually re-extracted again. So they re-extract the RNA from the positive samples. Those samples are sent to my lab in Aalborg in plates. So we get 96 well plates with positive SARS-CoV-2 samples in, set up in a format where we already have space for negative controls, positive controls, and everything we need. So we can just take a plate and put it through our factory up here in the end. And we run through 1,000 samples a day, so around 12 plates. And that's currently around the number of positive samples we have in Denmark. Addition to that, we have the government lab and a few other large labs in Denmark that are helping out when we surpass our capacity of 1,000 samples a day. So we've set up a factory, but again, we are very privileged in Denmark to be able to do this.
Cool. So I'm sure a lot of people, I mean, a lot of us here have questions. So we sort of skipped the introduction. So maybe if you guys have questions, maybe you could just start it with some introductions, like what your role is in the project.
Hello, good afternoon. Can I start, Khalil?
Yes, please.
So good afternoon, guys, particularly to Emil and Mads. Thank you for joining us today. Can you hear me, guys? Or am I speaking slowly? Okay. So I'm Michael Mitch. I head the wet lab team of PGC Mindanao. So just to give a brief background in terms of the situation which we are in, last year when the pandemic started, there was only one operational testing laboratory in the island of 24 million people. And that laboratory is even catering samples outside of Mindanao. So we established a training program to establish molecular diagnostic labs in different regions because there was a severe lack of personnel capable to do molecular diagnostic tests at the basic level. And for the first few months, there was quite a bit of resistance. So it was only until later, around 6 months after, where we were able to operationalize the training program, and we got the support of the government after quite some, convincing them and discussions. And then, so at the end of last year, we were able to train around 19 laboratories in the span of 6 months. So which most of them are already operational. I think 16 of them are already operational and contributing to the testing capacity. So now the effort of Philippine Genome Center Mindanao, because we were capacitated by Project Accessible Genomics. Previously, we are already doing sequencing work, but it was limited to Illumina, and we did not have any machine in our lab. We only do the live prep, and then we send the libraries outside or to Manila to have them sequence it, and then the data is just returned to us, and then we analyze the data. But we did not have any prior experience to actually running the sequencing itself. So when we had that capacity last year, thanks to the funding from Jogel, so we thought that why not we decentralize it for a more efficient or effective or a responsive surveillance system. So that's what we are trying to do right now. So we are also in the process. PGC Mindanao is also in the process of developing a training program, which is fit or tailored to the needs of the personnel landscape that we have here, because there's quite a difference in terms of their training and background, because most of the medical technologists in the Philippines and the laboratory analysts that are supposed to do these kinds of work do not have molecular biology classes integrated in their curriculum, and they are only starting this year. So that's the kind of challenge that we are trying to address. And hopefully, based on the we want to learn best practices. For instance, like um, protocol modifications and the considerations that uh, you are observing in your workflow, so that we'll be able to, as much as possible, replicate or have an idea of what are the things that we should look out for, considering the difference in the resources. So my first question would be, uh, are there any specific protocol modifications that you think would be beneficial when using the Arctic workflow compared to the standard protocol that was published in the site?
So I would say stick to a standard Arctic protocol. And the reason for that is that that's what everyone uses around the world. That means that you have a much easier way of getting support. So one of the things that Emil did in the start of the pandemic in Denmark was that he modified the protocol basically to introduce the barcoding step when you do the PCR instead of ligation. And that gives some advantages if you do this at really large scale, because you can barcode many hundreds of samples. But I would say for most practical purposes, you're not going to have that many samples available, and you probably don't have the funding to run at that scale anyway, because then it's not the sequencing that's the expensive part anymore, it's the reagents anyway. So I would definitely just stick to the standard, I would say the low cost version of the Arctic protocol. That seems to work very well for a lot of sites around the world, and you can get a lot of support internationally for it, also for the bioinformatics. Because once you start to tinkering with the protocols, you also start to need to tinker with the bioinformatics part of it. And that's often, in our experience, the most difficult parts to do. So in Denmark during the summer, we actually set this up at all the hospitals in Denmark too. But instead of introducing our protocol that we modified, we actually introduced the normal standard Arctic. And that was mainly because of the bioinformatics that you could use off-the-shelf scripts that you didn't have to modify. So even in Denmark, we actually choose not to implement our like high throughput factory protocol, but go with standard Arctic in the places you actually rolled it out to. In our mind, that has much more advantages comparing to trying to implement like a factory protocol as we run.
Yeah, I would agree with Mads as well. So we developed a really nice protocol and it's working really well. But I mean, if others should try and replicate it, I think it will require some training or at least a lot of time for optimizing it for your lab and so on. So I think the go-to will just be the standard Arctic.
Okay, thank you. Our next concern is in terms of sample availability, because with a lot of asymptomatic transmissions going on here, because the testing capacity it's still not that sufficient to really cover all cases as much as possible, and we expect that a lot of cases would be on the end of high CT values. So it's not at that criteria alone. It's gonna be excluded. So or we won't be able to get a whole genome. So in terms of the targets, because the labs are using different kits. There's no standardized protocol that's being implemented by our national government in terms of testing. So when we work with a lot of labs, so they're using different detection kits with different targets. So how in terms of selecting targets to be able to generate whole genomes based on the CT values, like do you have like any suggestion like which genes should we focus? Because some genes they have quite a tendency to have different CT values from the same sample.
So we're actually not involved in that part in Denmark. That's handled by the government. We only get the RNA afterwards. But also in Denmark, they use a different set of protocols. But if you have enough samples available, I would go with selecting samples with CT values below 30, although that can differ quite a bit from kit to kit. But once you start to get above CT 30, you also need to really be sharp on contamination in your lab. That starts to be something you need to pay a lot of attention to if you start to have samples with too little virus in. Even at our place where we have a lot of experience, we still struggle with contamination from time to time. So, but being able to select the samples based on CT and only working with high CT value samples, that should ease it a bit, because the risk of contamination is not that great, or the impact of contamination at low levels is not that big on samples if you already have a high virus load in them. So if you can, just work with things below CC 30 if you have enough samples.
Just a follow-up question, because we have like some sample types with low CT values, let's say for RDRP, and like very high CT values for N gene or E gene, like E gene, and we still don't have enough data to conclude whether RDRP as a marker is sufficient to like really in terms of a correlation of how much genome will we get. So do you have any suggestion for that?
No, because I'm not involved in the qPCR part of it in Denmark. We can try to ask some of the people who are involved in that part, but that's outside my scope.
Okay. All right. Thank you. I think I don't have any questions left. I'm still thinking. Maybe the others can ask. Thank you. Thank you so much for your feedback.
Khalil, can I go next? Is that okay?
Just go ahead, Omer.
Okay. Hello to both of you guys from Denmark. My name is Homer. I'm a volunteer bioinformatician on this project. My question would be, how did you tweak the pipelines in order to make it more, make it make it work better in your factory setting or high throughput sequencing activity? Because as for with your number of samples that you process, you should require a fair bit of computing firepower. So how did you make it work in your case? So yeah.
As I said, I'm very fortunate that we have more or less unlimited resources to play with. So one of the big problems in the start was having enough base calling power. So the nanopore produces a lot of data that needs to be base called. Usually we handle it by just sticking some powerful normal graphic cards in our computers. So we will buy a high-end NVIDIA card and stick it in the computer and base call it on that. But we quickly ran into problems because we run 12 to 18 minions in parallel. So we didn't want to have 12 to 18 computers standing in line. So we have access to a highly powerful GPU cluster using NVIDIA's high-end Tesla V100 cards. So we have a node with 8 of them. And what we do is we stream our data. So when it comes from the minion, we just take the raw FAST5 files and then we stream it up to the GPU nodes that do the base calling. And then we stream that down again. So we have like a whole extremely powerful GPU node to handle the base calling of the nanopore data. What we built for the hospitals in Denmark was we just took a gaming computer basically with a good graphics card, and we used that for base calling. And I think you can get that for a reasonable price, but that's our normal go-to. Stick a gaming graphics card in it and use that for the base calling. The way we run our factory is that we use 2 days in the lab to prep samples. So we receive samples early morning, start on 12 plates, so 1,000 samples. Day 1 is just doing the PCRs and cleanups. And day 2 is doing the nanopore prep and putting it on the sequencer. Then we sequence overnight. So normally we start the sequencer around 3 p.m. And then we have 2 options. We run around 100 samples per minion. And once we've generated 10 gigabases of FASTQ data, we trigger our genome workflow. Alternatively, if we have a bad flow cell or something like that, we trigger it at 6 a.m. the following morning. That means that we can have our data on our genomes the following day before 9 a.m. That means we can do rapid turnaround. So for us, at least, because we provide the data to the government, they need to have it in a predictable way. So again, the factory mindset that we trigger workflows both based on time and based on data availability, but then utilize very powerful GPU nodes to do the base calling. That's the main bottleneck we see, at least when we do high throughput data.
So for the 8 V100s that you have, those are provided by the government, or was it provided by
it's
your
provided
lab?
by my university. So
by the university.
Okay. Yeah, we have some GPU clouds, so they have a high focus on AI research, and part of that they have a number of these AI large GPU nodes. And I just asked, can I take one of that? And they said yes. So we have just temporarily taken over a part of the AI GPU resources at the university to do base calling instead.
So for those 8 V100s, how much data can it process, and how long does it take to say process, let's say 100
So I think
samples?
So we do just live base calling of it. So right away when the data is off the minions, it gets fired up into the AI nodes, and then it just gets live base called. So it can follow up in real time. We've run up to 24 minions in parallel, and it didn't struggle keeping up with that. And I would say if you have a normal powerful gaming GPU graphics card, you can keep up with 2 minions running live with high accuracy base calling. That's at least our experience. So if you buy a decent NVIDIA graphics card, you can do live phase calling of 2 flow cells at a time.
Wow. Nice. Thanks. That's all I was. That's all
If
I'm
I may
about to
ask
ask.
a
Yeah, so
follow-up
that's
question.
the
Sorry, go ahead.
yeah, that's one part of it. That's generating the raw data, getting the FAST5 files to the FASTQ. Then afterwards, we trigger our genome workflow, and in that we run the Arctic Medaka protocol. And after we have generated genomes using that, we chuck it into Pangolin and Nextclade to get genome assignments and also specific mutations, amino acid changes directly from Nextclade. And on top of that, we have some automated scripts running a few QC reports afterwards. And the whole workflow we run 200 samples in a batch, takes around 30 minutes on a 100 core CPU node.
Wow.
So,
That's a
but
fair
again,
bit of compute.
yeah, we are privileged. Again, we have unlimited resources, so we just set up the compute we needed, basically.
Yeah, that's an amazing setup.
Yeah, I mean, that's part of my one of my research interests. So all I could say is, wow, that's a fair bit of compute you have there. So thanks.
That's a special situation in Denmark that both my university and the government and the private foundations that normally found my research. I called them and asked, can I start to use my private funds that I have from my research grants to Corona research? And they said, yes, go ahead. Then suddenly I had like a big bank account and enough people to get going. And now we have support from the university and the government with basically endless funding to make sure we do that. So it's difficult to compare to the situation in Denmark. We are very spoiled here.
I still think you make a nice point saying like a good gaming PC will get you a fair bit. So if you don't need this complete factory settings, it will be way overkill with this.
Yeah, that's what we've done for the hospitals in Denmark. We just bought a gaming PC, and that works nicely.
I'm curious about your solution to the hospitals. Like, what's their unique need that they need to sequence themselves, and how is it different from like sending the samples to you?
So what we do is national surveillance in Denmark in my lab. So we are helping the government having an overall picture of which variants of concern and potential of concern are spreading in Denmark. On the hospital, they're more interested in reinfections of the patients, and maybe they have some small outbreaks. And they can do faster turnaround than we can do. In Denmark, on an overall scale, we have a turnaround of 5 days from positive samples to genome, which is still extremely quick. But locally, they can do day-to-day sequencing in the hospital. And also they can prioritize resequencing the same patients many times if they are sick over a prolonged time. So that's what they focus on. That's their patient and their local outbreaks, and trying to use that to take decisions there. And then our part is the national level overview.
Cool. Thank you.
Yeah, can I go next? So hi guys, I'm Ineke from the University of Amsterdam, and I have benign experience from in my PhD. Very interested in how we can use portable genomics mostly for wildlife health monitoring. So that was my role in this project, to help with the benign capacity building, so to say, share the knowledge. And also think about how, we can use all this new capacity after the pandemic one day, hopefully is less intense. So I think this was also partly of the conversation on Twitter, but I wanted to kind of get back to that I'm kind of curious. Do you wash your flow cells? Do you reuse your flow cells? Or, if you run so many samples on it as you do, is it just basically a one run and then it's depleted? Because personally, I've only used the wash kits from a few years ago, and I know they've massively improved over the years. So I'm just kind of curious, how much life you can get out of the flow cells with the amount of samples you run.
Yeah, maybe Emil can answer that.
Yeah, sure. So usually we can wash the flow cells and use them again, but we don't use the same flow cells for corona sequencing as it is now, because I mean we have so many samples and we're burning through so many flow cells each week. But we are saving some of them for, let's say, we have like off projects or other part of the university might be able to use used flow cell. And the wash kits are really nice now. So usually when we generate like easy 10 gigabytes of data from the flow cells, we can wash them and still have like 1,000 pores available. So that's super cool. I'm not sure how it will be with the standard Arctic. So the workflow we're using are using a bit longer fragments. So we're actually sequencing the coronavirus genome in like 1100 to 1200 base pair fragments. And the smaller fragments are a bit harder on the pores, but with the new chemistry and so on, it's really stable with the small fragments as well. So I really guess you could wash them and reuse them.
Yeah, so because are you at the moment using the R10 chemistry or a combination of both?
Yeah, so we're only using the R9.
R9, yeah, okay. Yeah. Okay.
So
yeah,
at least for COVID, yeah.
Yeah, and I guess this is also useful for, I mean, for in Mindanao, we already talked, I mean, Mitch, correct me if I'm wrong, but they're also really interested in One Health approaches and investigating viruses in wildlife and things like that. So it's a good reminder that we should use the flow cells wisely, but just wash them and store them so that they can be used for other projects in the future.
Yeah, I mean, for sure. I will just avoid using the same barcodes because there can be some spill left over. So as long as everything is barcoded and you're using different barcodes, it's fine.
Yeah. Cool.
Yeah, that's also what we do in the research setting. Often, even if you just have like a few hundred pores left, it's often more than enough data you can get off it if you're just doing some small tests. So it's really useful. The new, I say last year, the flow cells has become much more stable, and also the wash kits has become much better. So it really works nice, and you can do many washes and still have useful data out. For many cases, you don't need much data just to test a few things. And that's really valuable and saves a lot of money.
No, absolutely. In my personal research, I've focused on amplicons, and honestly, in 15 minutes, you've got enough data, maybe even already too much data. So there's so many questions you can answer with just a short amount of sequencing. And you're right, if you use different barcodes, then you can really make the most out of your flow cells. Cool, thanks.
Just to share in relation to Nika's comments, we're actually working right now in a One Health surveillance for coronaviruses. So this year we're gonna do extensive sampling of wildlife from bats to non-violent mammals, livestock, and even humans in wildlife livestock human interfaces. And we're currently trying to design a panel for pan-coronavirus enrichment, and hopefully we can apply the same protocol the Arctic protocol for the sequencing using nanopore. So that's also something that we are trying to work on right now. Yeah.
Hi. Can I go next?
Sure,
To
good.
ask yeah. Hi. Hi, Emil and Mads. It's good afternoon from here. I'm Paul. I'm the bioinformatician of Philippine Genome Center Mindanao. Yeah, I'm also the head bioinformatician of this project, Accessible Genomics. So maybe just one question. Is there like a correlation between the CT value? So for example, the lower the CT value is, more the genome you could cover, like maybe like in your experience, have you had like like that correlation, or for example you have a lower CT value, maybe in your case you have low CT values, then you're able to assemble 100% complete SARS-CoV-2 genomes, or at least almost complete SARS-CoV-2 genomes that are that you could confidently assign to certain lineages, like so that because maybe one of the goals here is in this project is that we're able to identify the variants that are circulating in the area, so that we're able to get a picture of the circulating variants, and that would tie into like the management of the pandemic here. So
Yeah.
I
So
hope right here.
yeah, sure. So again, this is dependent on the qPCR setup you have. But in Denmark, with the setup we have, we can see that everything with a CT value below 32, we expect to make a perfect genome on. That's what we can see from the data. From 32 to 35, it starts to be more difficult. I would say we have a success rate around 50% of getting a high quality genome from that. Above 35, it's very difficult. And we have a success rate of maybe 10 to 20% at max. So I'd say that's like guiding posts for what we see given the test setup we have in Denmark with the qPCR. Below 32 seems nice. 32 to 35, it's a twilight zone where it gets difficult. Above 35, very difficult to get useful genomes out of that. One thing you could consider, so you're focusing on whole genomes. In Denmark, we actually do a mix of many things. So again, we are privileged in Denmark. So we, of course, sequence everything with full genomes. But because there is this 5-day turnaround, we want to start contact tracing early in Denmark, as early as possible. So we do have qPCR setups that detect the different variants of concern. And we also have what calls spike sequencing. So we sequence a single amplicon within the spike gene using Sanger sequencing, actually. And that's a single PCR reaction using the same reagents as for qPCR. You just take that product and you ship it to Sanger sequencing instead. And there's no barcoding, there's nothing, you just ship a plates and then get the results back. That's a very easy way to actually monitor and very cheap way to monitor the variants circulating. Of course, we would love to have complete genomes, but in Denmark, to improve the speed of being able to start contact tracing, because we want to keep out some of the variants of concern, we actually do this. And I think it might be an option in other parts where you can very cheaply and very simply in the lab, you can monitor a part of the spike protein that is essential for all the variants of concern. It's much more simple in the lab to run. It's like one PCR. No barcoding, nothing at all. Bioinformatics is also extremely simple to do. So it might it's not as sexy as producing whole genomes, but I think you get all the benefit for a fraction of the price and a fraction of the complexity.
Yeah, and I think I would just like to add for the lab experience when doing these whole genome sequencing and CT values and so on. So generally the Arctic protocol and our protocol is quite robust. So if you have samples of, let's say, CT30 or 28 that fails, but in theory should work, our experience is usually that if we redo them, they will fail again. So the protocol is quite robust. So I mean, sometimes we redo samples if some funky things have happened, but in general, if samples fail, they will fail again.
Just want to clarify. When you meant that the spike protein can be used for variant monitoring, so is my assumption that the phylogenetic signals that can be obtained from that region alone is like sufficient to assign it to a particular lineage in comparison to using whole genomes? Because that's also one of our main concerns, because we expect many of these laboratories won't be able to, like if we are going to run per batch, so it might take several days for them to accumulate like the number of the minimum samples, let's say 24 with good CT values before they're able to run that. So for us to maximize utility of the sequencing as much as possible, we also want to get as much information, even if we are dealing with low quality samples, like let's say high CT values. So is my assumption correct on that aspect, Mads?
I'm not sure I fully got your point, but I think full genomes are nice if you do genomic epidemiology. But I think a lot of the benefit, for example, surveying the new variants out of India or the V117, you can do that by just tracking mutations of concern or mutations of interest. You don't need the full resolution to figure out which subtypes are actually circulating. So depending on your purpose in your country, it might be more important to be able to overall track how many of V117 do you have, how many of the new Indian variants do you have, instead of being able to go to full resolution. So it really depends on the needs of the country. I would say the thing is that if you only do like sequencing of this small part of the spike protein, it's a bit more difficult to share internationally right now because we don't have a way to report that. And that's the nice thing about doing the genomes, because we have a system in place that can handle that. But I would say in a large part of the world, it would probably have more benefits to just focus on the spike right now, at least until we start to get more evidence that other parts of the genome should also be used to do these inferences. But right now, we can see that all the variants of concern can easily be distinguished based on a relatively small part of the spike protein. Of course, we cannot get all the details that we would love for detailed investigations, but right now, maybe it's not the time for the detailed investigations.
Mads, would that mean that the sequencer could produce more, or could sample, or could handle more samples if you are just like sequencing the spike? Is that correct?
Yeah, I think that the practical limitation there is getting enough barcodes to be able to utilize that capacity. At least I'm a little concerned about washing and reusing it with the same barcodes. I think that's a bit too uncertain, but the setup here is more suited actually for Sanger sequencing. And that's still something that's actually widely available across a large part of the world at commercial places very cheaply, normally with very fast turnaround. So the places that do it in Denmark actually just ship it out to commercial providers, and then a few hours later they have the results. So it's we should figure out what is the right tool for the questions we want to answer. And I'm a big fan of nanopore sequencing and whole genome sequencing, but sometimes there might be tools that are actually better suited to get information fast.
Yeah, I would like to follow up on that, because I do think that that's true. Like, it's also, I talked to, I took the MinION to Indonesia and trained some veterinarians there, but we also discussed, like, you know, when will you want to use the MinION, or when is a normal PCR with a gel sufficient? You know, if you can answer some questions with that, then don't go ahead and continue with MinION sequencing just because you can, if you already have an answer to your question. It's just that this is a question for the rest of the team in the Philippines. Like, how is it there? Because I know that, for example, in Indonesia, it was really difficult to even send samples from one island to another island for Sanger sequencing. You would need a lot of paperwork, and then it would be very slow, definitely not the same day that you would get the results of your Sanger sequencing. So I guess this is why I think what I know that in some countries is a bottleneck. So I'm kind of curious, like, for example, Mitch or someone else, what's the situation?
It takes 2 weeks. We usually ship before we usually ship to Macrogen Korea. So the fastest the results can return to us is 2 weeks. Now we also sometimes you also ship to the Philippine Genome Center in Diliman or in Manila, but right now they only have a very limited staff, and they're focusing on SARS-CoV-2 biosurveillance. So their priorities are shifting all the time. And it also takes, like, you know, almost 2 weeks for them to run on samples.
Yeah.
Unless you have connections that can fast track the analysis for you.
Yeah, okay.
You mentioned, Mads, that the bottleneck will be the number of barcodes. And then you also mentioned in Twitter, because I'm the least I have the least scientific background in this call. So, but I suppose other people watching this might also find it helpful. You mentioned in Twitter that you mentioned, like, just take your primer and add 10 random NT. What does it mean? I have no idea what that means.
Yeah, maybe Emil can fill in here. This is his normal line of work, that's developing assays like this.
Yeah, so I'm not quite sure of the question.
Yeah, I asked on Twitter, like, how are you doing this 96 times 4 barcodes per flow cell?
Oh, okay. Yeah. So this has been a lot of work actually designing using these barcode sequencers. So there are some tools available where you can like develop these sequences and make sure you can actually separate them bioinformatically and you won't have bleed over and so on. So the limit would be depending on the length of your barcode. So if you need a lot of barcodes, you will need to increase the length of the barcode and so on. And I mean, I think for Illumina sequencing, it's not that big of a problem because you have a really high accuracy, but on the nanopore, you need to differentiate the barcodes a bit more to take into account that maybe errors that will get base called incorrectly. But yeah, I mean, for our setup, we have developed these like 4 times 96 different barcodes. That should be good to go.
Yeah,
How do
how
you-
do we do it in practice, Emil? How does a primer look like with barcodes on?
So, I mean, I have something I can show you.
And also the question is like, is it possible for us to actually do that here?
I mean, the entire PCR barcoding setup. I mean, I think it would be possible, but again, I think it would be the purpose of actually just staying to the standard Arctic protocol, because yeah, this is developed for like the PCR setup. So yeah. I guess.
Yeah, then you would need to do your own custom setup, and I think that's a bit too difficult to actually implement at scale, because then you also need to do the custom bioinformatics at the same time.
Yeah.
So to me, I would just stick with the nanopore setup that already exists. But in principle, it's easy, but it requires you have experts in every place because it needs to be custom solutions that's implemented.
I think, can you see my screen now, maybe?
Yeah.
Yes, yeah, we can see.
So this is just like a brief illustration of the workflow we're actually using. So this will be the standard Arctic primers in the 1PCR, and this will be like a generic sequence overhang in the 5' end of our primers. And then we get all these amplicons. It's actually just the standard Arctic amplicons, but with these generic sequences in each end from both pools. And then we do like the PCR2 setup over here, where we have these different barcodes. So we will just target the generic sequences we added to all the amplicons, and then we have like, yeah, these 96 different barcodes. Actually, yeah, we have 96 times 4 different barcodes. So then we can add a small barcode sequence to all amplicons as well. Doing this 2-step PCR instead of the ligation setup. It's, yeah, I mean, it works really neatly, but again, you'll have to change a lot in the bioinformatics, and you'll have to change a lot in the laboratory workflow. And I mean, for support, we will likely be one of the only laboratories that are actually doing it like this. So yeah, I think it's neat for our setup, but yeah, that's something I recommend doing without doing a lot of time of development and
normally run
optimization.
Illumina setups. You do you use the barcoding in the PCR, where in nanopore you could do the barcoding in the ligation instead.
Khalil, so if you are to pursue this, Khalil, it will require you to design a custom panel like with IDT to modify their primers or really design new primers with these overhangs. So it's quite really
Exactly.
tedious.
And costly.
Yeah, I mean, it will require optimization, because I also mentioned we're doing these like 1200 base pair amplicons instead of 400.
Yes.
Because in our PCR, we will have like only 2/3 or 1/3 of the products that you will have in the standard Arctic, which will complicate the PCR even more. And yeah, I mean, I think it will be the purpose. I really do. I wouldn't recommend this unless you will make this huge factory
And
setup.
the cost will also exponentially increase because the benefit with the Arctic panel right now is there are many users. So if you are to design a custom panel, the cost is just really very big.
All right. So this is really like building a factory versus like renting or just buying off the shelf.
Yeah, and in this factory, we have all the experts that are designing this right on a day-to-day basis. I think the Arctic is really awesome. A lot of people is using it, so I would really stick to that. Unless your governments in the different countries go in and say we want to make a factory, then I think you should think in these lines.
All right, it's clear now.
So
Okay,
the
thank you.
standard Arctic is at like 24 barcodes right now, or is it 96?
There's a the old one is a 24, and then there's a new one with a 96. So we're hoping, but it's much more expensive to get 96, but on a per sample basis, it's much cheaper. So what yeah, we're hoping to get that next. Any other questions?
No, not really. Just that, yeah, I think the two-step PCR has been what I've been doing with my amplicon sequences. So yeah, it's really the same. So it's also already something you can do with the existing ONT barcodes. You don't even have to design them yourself. So yeah. Oh, and there's a question in the chat. I will read it from Joan. It's by how much does the barcoding system reduce the 5 to 15% error rate of the MinION system?
So, do you want to take it, Emily?
No, I mean you can as well, but I mean, so generally the barcode system doesn't do anything for the raw error rate of the nanopore system.
Yeah, so that gets fixed when we do the consensus calling. Afterwards, you pile the reads with errors on top of each other, and then you take the consensus, and then you have basically perfect genomes. The coronavirus genomes is by chance, that's easy to sequence with nanopore. So that's nice.
Cool.
The error rate, I think, is only dependent on the chemistry and the pipeline.
The biggest impact right now is actually the base calling algorithms. But right now it works nicely, so it doesn't need to get better than it is now.
Yeah, but just going forward for the possibly bringing this to some labs here. I'm just really interested about the solution for hospitals. If you have any resources that we could read on and see if we could use it here, what questions could hospitals answer with sequencing. I'm particularly interested in the actual presentation, pitching it to hospitals.
And that's the most difficult part, to be honest. And what we also struggle with in Denmark, that's getting them. What can you actually use it for, and what makes a difference practical on the in the hospital? And I think we are still struggling a bit in Denmark to figure that out. Of course, they're using it to understand if they have a local outbreak between, let's say, different patient wards, that they're actually infecting each other there. But I think it's a rare case that they don't already know that when they start to sequence. So from our perspective, the most useful thing has been on a national scale, being able to track the variants. And in Denmark, we used it when we spotted B117, we spotted that in mid-December. And we actually imposed much stricter restrictions in January in Denmark. So even though cases were dropping, we could see in the data that B117 was rising. And that meant the government actually imposed much stricter restrictions, where other countries were loosening it. In Denmark, we actually kept hard restrictions. And that meant we hopefully now didn't get the third wave that we've seen in a lot of countries around us. I think that's been the main advantage in Denmark, being able to spot it on a national level. It's been much harder, even though Denmark is one of the countries where you think we could really use the data. It's been really hard because there's so many people at other places that needs to understand what you can use this data for. And often they want actionable data in real time. And even our world-class 5-day turnaround is often too slow to really make it useful. And that is just a challenge, I would say, that we don't solve in this pandemic. So right now, I think the most important part is having a global overview in the world of which lineages are circulating. I think that's also justification enough. Getting it really to make a difference at the individual hospitals, I don't think that is viable, to be honest.
Okay.
But it's extremely important that we have this global overview of variants.
For local governments, another question is like how much sequencing do they need to do for it to be useful for them? It's like, what's the minimum they need to produce, like for a city, for instance, or for a small region? It's like, what's the advantage that you could present to them, and what would it take for that advantage to come about?
So you were breaking up a bit, but the local advantage again is we have some variants of concern now that actually might change the restrictions you need to enforce in the society to keep the pandemic at a sustainable level. At least that's what we use it for in Denmark primarily. That is actually to make sure that we don't get caught off guard by new variants and we actually enforce restrictions too late. So that's what we're doing it, using it for in Denmark, trying to match the level of the restrictions in the society to the threat of the current variants that is circulating. But again, Denmark is unique because we have the capacity to sequence so much that we actually can go in and do this more or less in real time and pick out specific variants that we don't want to have in the country.
Yeah, so
I think it's very optimistic to think that this could make a difference on the local hospitals, but I think it's essential for the world that we track the variants.
Cool. And then maybe this didn't get through in my question earlier, but like if I'm asked by a local government, because if we want to help them set up a sequencing operation in their lab, like what's the minimum number of sequences or samples they need to produce for it to be useful for them?
It depends on how much they test in the community. I would say so. You can't distinguish the number of PCR tests from the number of genome sequences you do. So it depends on how much you have of a pandemic. But I would rough estimate a few hundred sequences per week would be nice. If you can only do 10 to 20, that's also good. Then focus on getting representative samples as much as you can instead, and put your effort there. Then that would be fine. 24 a week would be great if they're able to do that. That are then not just one outbreak, but spread around in different parts of the local community.
And then how do you suggest the analysis be made? Is it, or at least in your case, it's your team that analyzes data and gives recommendations to the government? And do you see this in other cases as well? Like for instance, if there's not an if there's go.
So we don't advise the government. The way it's set up is that we collaborate directly with the national lab, and it's then the national lab that then advises the government. So we help by setting up the analysis and the pipelines and the reports for the national lab, but then it's the national lab that has the connections directly to the governments, and you need to have that. Without that, no one is going to listen to you. So that's difficult. At least if you come and say now you've set it up, you need to have the government support to make sure they actually look at this.
Yeah.
But having a weekly report of the variants of concern that is circulating in the community, I think would be very valuable to figure out if there's something that might potentially have an impact. But again, I think if you are in a country with limited capacity, it's much better just to track the number of infected people, so increasing the PCR testing capacity. So it's difficult, and I'm not sure how big difference genome sequencing can do. It's important for the world that we keep track of the variants. It might be a bit more difficult for the local authority to figure out what it actually could be used for. And I guess that's the big difficulties, because it's on a local scale. You need to implement it
for the globe. Okay. All right. So, hey, thank you so much, Mads and Emil. This has been very helpful. So, so many things to think about that we talk about later with the team. And yeah, so thanks again, and hope to see you around in this space.
Yeah, sure.
thank you,
It was
Mads.
All right.
It
Have
was
a
nice
great day. Goodbye.
hearing what you're doing. See you.
Thank you.
Thank you. Goodbye.
Thank you
Bye.
so much, guys.
Thank you.